Hello, my name is Paige, and today I will be presenting on the CRISPR-Cas9 system, a gene editing tool for crops. What is CRISPR-Cas9? CRISPR stands for clustered, regularly interspaced, short palindromic repeats. Cas9 is the CRISPR-associated protein 9. Pictured on the right are Emmanuel Charpentier and Jennifer Doudna, the pioneers of the CRISPR-Cas9 system. Their research started out studying the immune systems of Streptococcus pyogenes. In the bacteria's defense system, they found a protein, the Cas9 protein, that was responsible for cutting and dismantling foreign DNA. What they couldn't figure out was how the protein found the DNA. It wasn't until Charpentier's discovery of tracer RNA that brought together the CRISPR-Cas9 system. Together, Doudna and Charpentier developed the CRISPR-Cas9 system in 2011. Their discoveries earned them the Nobel Prize in Chemistry in 2020. So how does it all work? Tracer RNA and CRISPR RNA are fused together to form a guide RNA. CRISPR is programmed with a specific DNA code. CRISPR will then locate the matching code in the DNA sequence of the cell, and the Cas9 protein will make the cut. The DNA can repair itself by a process known as non-homologous end joining, NHEG, shown in Part A of this diagram. This repairing process leaves out the genome sequence that was cut by the Cas9 protein. Another method of repair is known as homology-directed repair, HDR, shown in Part B of this diagram. In this repair method, a template with a different DNA sequence can be inserted into the DNA to change the genome. The benefits of CRISPR versus other gene editing tools is that it is inexpensive, user-friendly, and is life-changing technology, literally. Because this technology is still fairly new, there is, of course, room for improvement. However, the CRISPR-Cas9 system has proven to be successful in editing the genomes in plants. The CRISPR-Cas9 system can potentially replace current gene editing technology, such as zinc finger nucleases, CFNS, or transcription activator like effector nucleases, TALENs. CRISPR-Cas9 could even replace genetically modified organisms, GMOs. These previously used techniques are more complex, expensive, and less efficient than the CRISPR-Cas9 system. Researchers have discovered that editing the genomes in plants can lead to improved traits such as better yields, resistance to abiotic stress, resistance to pathogens, and herbicide tolerance. First, researchers must identify which genes are responsible for, let's say, the amount of fruits a plant can produce. Once the gene has been identified, it is programmed into CRISPR, and it will lead the Cas9 protein to that part of the sequence in the plant's DNA. By altering or removing the gene completely, the plant will be able to, in theory, produce more fruits. But why is there a desire to produce a higher yield in crops? By 2050, the human population will grow to be greater than 9 billion people. With an ever-growing population, there will be an increase in demand for food. There must be a way to compensate for the higher demand. As you can see in this chart, countries like India and China will have to continue to provide for more than 1 billion people. In order to effectively provide food for this large amount of people, there will need to be a mechanism to increase crop productivity. Along with a growing population, crops will need to adapt to a changing climate. Abiotic stress like drought and extreme temperatures make it difficult for crops to thrive. This may affect their ability to produce high yields or even survive. As climate changes, crops will need to adapt to an environment they would normally not survive, and researchers have discovered there might be a way to help. By editing specific genes, crops can adapt to a changing climate. By altering the genome of plants, researchers have been able to identify the genes that are affected by undesirable conditions, cut the gene out of the DNA sequence, and allow for the plants to produce better crop yields and adapt to the changing climate. Drought, extreme temperatures, oxygen levels, and soil toxicity all affect the productivity of crops. One study shows the knockout of the PYL1, 4, and 6 genes produced a high heat tolerance for rice. The addition of ARGOS8 gene in maize produced a higher grain yield in drought conditions. In soybean, the knockout of DRB2A and DRB2B resulted in a tolerance to drought as well as salinity. The removal or alteration of these abiotic stress-related genes allow for the crops to adapt to environmental factors such as drought and high temperatures that would normally affect the productivity and lifespan of crops. Pathogens, like bacteria, viruses, and fungus, can affect plants just as they do humans. Researchers have discovered that by disrupting S genes in plants, it helps build an immune response to bacteria, viruses, and fungi. The CRISPR-Cas9 system is used to minimize the effects pathogens have on the plant's ability to survive. The OSERF922 gene disruption was shown to prevent blast lesions, a common fungal disease in rice. The disruption of SLPMR4 in tomato resulted in a partial resistance to mildew. The disruption of these S genes will enable crops to build an immune defense against common diseases and also prevent the spread of disease amongst the crops to ensure a higher level of productivity. Herbicides are used to control weeds that compete with the crop's access to water and nutrients. Herbicides have the ability to bind, which means the crops are absorbing these harmful chemicals. CRISPR-Cas system is used to create a tolerance against herbicides, used hoping to only affect the surrounding weeds. The editing of the ALS and EPSPS genes have shown that they enable the plants to resist the binding of the herbicides. This creates for a healthier crop, free of herbicides, and can help with an increased production and lifespan. Editing the nutritional value of crops can be beneficial to humans. People who are gluten intolerant or malnourished can benefit from these studies. The knockout of alpha-gliadine reduced the amount of gluten found in wheat. The knockout of ZMLPK reduces the phytate content, which helps aid in digestion. And the disruption of the FAD2 gene increased the amount of oleic acid found in oilseed crop. Being able to genetically alter the genes of plants to produce a higher nutritional value is important to human health. With an increase in crop productivity, along with an increase in nutritional value, the CRISPR-Cas9 system proves to be an effective way to support an increasing population. Here are some specific examples of CRISPR-Cas9 system being used in plants. In tomatoes, the knockout of the SLMPK20 decreased the amount of sugar metabolized in the fruit, which gives it a sweeter taste. The knockout of SLAL-MT9 decreases the malate content, which gives fruit a tart taste. SLER, SP5G, and SP genes were edited to alter the length of the tomato stems and the flowering process. After editing nearly 140 different lines, researchers saw improved traits such as larger fruit size, increased number of fruits, and higher vitamin C content. This proves that CRISPR-Cas9 has the potential to develop crops with high yields and better nutrition. Another application of CRISPR-Cas9 gene editing was used in rice crops. The knockout of GW2, GW5, and TGW6 resulted in larger grain size and weight. Mutations of OSHD2, OSHD4, and OSHD5 allowed grains to mature early. The mutation of CLVTA3 increased the number of seeds and grain weight when compared to wild rice. This will aid in crop productivity and will be beneficial to an increasing population. The study of CRISPR-edited rice has been mentioned numerous times throughout this research because rice is a staple crop in many countries. These improved traits in rice may be crucial to a larger population in the future. So what does this mean for the future? Because of an ever-growing population and constant climate change, the CRISPR-Cas9 system can help food insecurity in countries that rely heavily on crops, like rice grains and wheat, for survival. By increasing nutritional value, greater production, and adaptation to changing climates, this can ensure crops' ability to provide for an increasing population. This system has proven to be effective and efficient, and is moving forward to the next step in cultivating disease-resistant and climate-tolerant crops around the world. Thank you for listening to my presentation, and I hope you enjoyed it.